How the gut and the brain communicate has fascinated us for centuries. The gut-brain axis transforms information via the vagus nerve from food to feelings. Once eaten, digested food particles enter the small intestine, which is covered with a velvety layer of villi. Each villus is lined with a single layer of epithelium. This layer is made up of different cell types. One of them, the enteroendocrine cell, is unlike the others. It is our gut sensor. In addition to communicating through hormones, we discovered that enteroendocrine cells also synapse with nerves, including the vagus nerve. We call those enteroendocrine cells synapsing with nerves neuropod cells. They sense and react to their environment. They sense mechanical, thermal and chemical stimuli such as nutrients or bacterial byproducts in the gut lumen. Inside neuropod cells, signals from stimuli are converted into tiny electrical pulses. These pulses propagate via synapses onto the afferent neuron of the vagus nerve. Vagal neurons carry the sensory information to the brainstem, linking the signals generated inside the small intestine to the brain. The neuropod cell connection with the vagus nerve serves as a conduit for food in the gut to influence brain function within seconds. This connection is also a potential portal for gut pathogens to access the brain. This new knowledge is a foundation for designing therapies to treat disorders related to altered gut-brain signaling.